This is Group 11. We are going to explain about MHC Class 2. There are several types of MHC, and today I will be comparing MHC Class 2 and MHC Class 1. Both of them have peptide binding sites and they are on cell membranes. First, the basic and common role of MHC is to present peptides made from broken proteins to T cells as antigens. This is the common role to both Class 1 and Class 2. The difference between Class 1 and Class 2 is where the antigenic proteins are obtained from and where they are presented to. In Class 1, the antigen made from the cell is broken down into antigenic peptides. And together with MHC Class 1, the peptides are presented to killer T cells or CD8 plus T cells. In other words, they present what they have been infected with. For example, viral proteins which are produced in virus-infected cells are presented by MHC Class 1. And also, they are working for cell-mediated immunity. On the other hand, Class 2 peptides are taken up by endocytosis. Antigenic substances made outside of the cells are broken down into peptides. Then it is presented to helper T cells, or CD4 plus T cells, together with MHC Class 2. In other words, it presents what it has eaten. For example, toxins and bacteria are presented by MHC Class 2. And also, they are working for antibody-mediated immunity. This is a table which shows the differences between MHC Class 1 and MHC Class 2. The antigenic peptides of MHC Class 1 come from inside of the cells, like viral proteins, and go to killer T cells. On the other hand, the antigenic peptides of MHC Class 2 come from outside of the cells, like toxins and bacteria, and go to helper T cells. All nucleated cells have MHC Class 1 on their cell membranes. MHC Class 2 is known to be carried by antigen-presenting cells, such as dendritic cells, macrophages, and B cells.
Next, we explain more specific mechanism of Class 2 antigen presentation. First, extracellular proteins, such as bacteria and toxins, are captured into antigen-presenting cells, and then they are broken down into peptides by primary lysosomes. Class 2 molecules, by the way, are formed in the endoplasmic reticulum with invariant chain. Invariant chain, or its fragment, creep, serves to prevent other peptides from binding to Class 2. At the end, peptides and MHC2 meet, get together, and move to the cell surface for antigen presentation. This is how MHC2 present antigen like bacteria and toxins. Thank you for your listening.